you guys hear me? Okay, good. I'd like to thank everyone for coming today for today's research seminar, Protocol Development for Vehicle Emission Toxicity Testing for Particulate Matter. My name is Pat Wong, and I was the contract manager for this project. I am an air pollution specialist at the Health and Ecosystem Assessment Branch. If you have any questions regarding this project, my email is here on the first slide. Just a couple of announcements to begin. For those of you online, thank you for joining us today's seminar. Questions for the speakers for after the seminar can be sent to the email you see on the screen here. Additional information about the speakers, such as the slides and expanded biographies, can be found at this site down here. Now, I have a number of housekeeping announcements for the attendees here in the room. Please look around now and identify 2 exits closest to you. In some cases, an exit may be behind you. In the event of a fire alarm, we are required to evacuate this room immediately. Please take your valuables with you and do not use the elevators. While staff will endeavor to assist you to the nearest exit, you should know that you may find an exit door by following the ceiling-mounted exit signs. Evacuees will exit down the stairways and possibly to a relocation site across the street. If you cannot use the stairs, you will be directed to a protective vestibule inside a stairwell. Should we have to relocate out of the building, please obey all traffic signals and exercise caution crossing the street. Okay, and I guess I'd like to just start off with a brief introduction. It is pretty well known that exposure to PM, particulate matter, or the toxicants on PM can give rise to a lot of different types of health effects. However, the actual mechanisms for these health effects isn't really known right now. However, consensus gives us a few pathophysical pathways which are generally accepted. One way PM or PM-associated toxicants could cause health effects is through DNA damage, which could lead to cancer. Another way is causing inflammation, which could lead to cardiovascular disease and inflammatory diseases or neurotoxicity. And a major step that many people are thinking of for each of these pathways is the formation of reactive oxygen species, which will increase oxidative stress, and that feeds into both of these pathways. Now, when the pathophysical mechanism of actions are known or speculated, in vitro assays, or basically assays that can be done in a cell or petri dish, or even chemical assays, can be used to compare the relative toxicity of samples with regards to a particular mechanism. Now, there are some advantages of using in vitro type assays. They're relatively inexpensive, especially when compared to animal-human studies, and they can provide rapid information about relative toxicity, potentially telling us about red flags or pathways for further investigations. Now, there's some challenges involved with in vitro toxicity testing. The major challenge is the interpretation of data. There are very few studies that can directly relate in vitro tox results to health effects. If a sample has like 10% or 20% more causes oxidative stress 10 to 20% more, we can't relate that to health effect. However, that is changing right now. There have been some studies, epidemiological studies, where people have measured oxidative potential of PM and associated it with increased asthma and cardiovascular related emergency room visits. And the oxidative potential is a better predictor than PM mass itself. The other issue with in vitro toxicity testing is standardization. Everyone basically uses the same assays. Same literature, same mechanisms, but each lab, of course, runs things slightly differently. A few years ago, and this is how this project kind of got started, we got data from 2 or 3 different labs for the same assay, but there were some inconsistencies, and we were wondering what were the differences. And the differences wasn't with the assay. We noticed it was how they were preparing the PM, and we decided to drill down on that question: does PM preparation for these assays affect the results? Now, the main question of this project was, how do sample preparation methods of PM samples affect the in vitro toxicology results? And what we wanted was someone to prepare the same PM sample using many different preparations and then submitting these different preparations to a battery of in vitro testing. And the reason this is important for us is this might form the basis of a standard operating protocol that we can give to future researchers, or maybe even give us an idea on why assay results differ from one another in different laboratories. Now, the PI that we selected for this project and will be giving the talk today is Dr. Keith Bine. He's a Bachelor of Science in Physics and Chemistry from Cal State University Chico. He He has a PhD in atmospheric sciences at UC Davis, and he's currently an associate professor researcher at the Air Quality Research Center and a research professor at the Center for Health and Environment at UC Davis. His two co-investigators are Dr. Norman Cotto. He has a bachelor's degree in biology from UC Riverside. He has a PhD in environmental health sciences from UC Berkeley and is currently an adjunct professor in the Department of Environmental Toxicology, and most people here know him as a staff toxicologist at the California Air Resources Board. Dr. Chris Vogel has a degree in toxicology from the German Society of Pharmacology and Toxicology. He has a PhD from Heinrich Heine University in Dusseldorf, and like Keith, he's also a research professor at the Center of Health and Environment at UC Davis. Now, before I turn the talk over to Keith over here, this is just a brief overview of the task division for this project. Starting with either a filter sample or an NIST standard, Keith developed a number of different protocols for extracting PM. He came up with 6 different ones, and he will be giving a talk about that. He took these preparations and gave them to Chris Vogel, who ran cellular response assays for inflammation and aryl hydrocarbon receptor activation. He also gave a set of these preparations to Dr. Cotto, who ran the oxidative stress assay, the DDT assay, and also did assays for mutagenicity, the Ames assay. So I guess they will talk about the results now. Keith?
Thank you, Pat, for that wonderful introduction and for framing the research that I'm going to be talking today about. And thank you, everybody in the audience, for coming, and anybody that's out there online. All of my gadgets work. Anyway, Pat already told you the topic and gave an introduction to me, Keith Bine, PI, as well as my co-investigators, Chris Vogel and Norm Cotto, who are out here in the audience. I want to start by kind of just framing the research and giving the bigger overall picture of the questions we're trying to address. First and foremost, the emissions and exposure landscapes, collectively referred to as the exposome, is constantly shifting. It's constantly changing. And why is it shifting? There's a lot of drivers. You have advancing technologies such as new emissions controls or new fuel blends or other kinds of technologies aimed to reduce emissions or to change the composition of those emissions. You have changing practices at the individual level all the way up to industrial or society level changes and practices. There's shifting regulations as we learn new things about the toxicity of different components as a function of chemical composition, as a function of size. Our regulations shift. We have new and emerging mitigation strategies to help us protect people from being exposed to things we know to be toxic. All of these things together are constantly driving changes in our emissions and exposure landscapes. So this gives a constant need to evaluate or to assess how these changes are affecting human exposure and whether or not the intended impact they are meant to have is actually happening, or if there are other unintended impacts that are occurring at the same time. So our main impetus here for controlling emissions is to protect human health. So how do we assess whether or not we're protecting human health? Well, traditionally, we do it epidemiologically, which is correlative, or we do it toxicologically, which is causative. So if we do it causative, mechanistically, through toxicology, what biological system do we use? Do we use an animal model? And if we do use an animal model, do we use human, ape, murine, zebrafish? What do we choose? If we do a cell culture, what cell line do we choose? Do we do it in a petri dish? Do we do an air-liquid interface? How do we expose the cell line to the sample of interest? New things coming up are organoids, little organs grown in a dish as a way to move away from animal studies and towards different paradigms of exposure. And so once we choose a biological system, how do we actually expose? And at what dose? Do we do an bolus exposure, intratracheal instillation, or pharyngeal aspiration? Or do we do a real-time inhalation where the animals are put in an exposure chamber and they're actually inhaling. If we do some kind of bolus, what dose do we do? Do we do a realistic ambient level dose? Do we do a highly concentrated dose? Do we do an acute exposure? Do we do just a one-time insult to their system and watch how it responds? Do we do a chronic exposure, which is more representative of what a human is actually exposed to on a daily basis? And for, cell lines, like I said, do we do injection or do we do deposition at the air-liquid interface, which is more representative of what's actually happening in the lung? Once we decide on how to expose, how do we actually collect the sample to expose them? Do we do field sampling? Do we go out into the ambient atmosphere and collect particles on a filter, bring it back to the lab, extract those particles, and then do the exposure? Do we try to take the animals out into the field and do real-time exposures next to a freeway or in a heavy polluted area? Do we do laboratory-generated aerosol, a dynamometer study or an engine? So exactly how do we collect our sample for the exposure studies? Once we collected our samples, how do we extract it? How do we prepare it? Do we do an exhaustive where we want to bring everything we possibly can off of that filter and include it in our exposure? Do we want to go after specific compounds in that mixture and test the toxicity of those one by one? Once we decide on our sample preparation techniques, what endpoints do we test? And at what time points do we test those endpoints? What system do we want pulmonary, cardiovascular, neurological? What endpoints? Inflammation, oxidative stress, mutagenicity, carcinogenicity. So all of these are variables in our effort to assess the impact of our exposome on human health. And each variable that you pick is going to affect the outcome that you witness in your toxicity testing. So basically what we're trying to do is trying to understand how varying one of these variables affects the outcome. So like any good experiment, we have to hold all of our variables constant, and we got to pick one variable that we can wiggle, and then we watch the system respond to that variable. So that's what we did. Can't see that, that's horrible. Okay. You're supposed to be, so what we did is we're going to do a toxicological studies. We're going to use a cell culture. We're going to do an acute exposure at a concentrated dose. And we're going to use laboratory-generated aerosol from a chassis dynamometer study. And we are going to do both exhaustive and selective particle extraction to sample preparation techniques. That's our variable. That's what we're going to wiggle, while we hold everything else constant. And we're going to look at a bunch of different endpoints. We're going to look at inflammation, oxidative stress, and mutagenicity. So we're going to do a single study, a single type of PM, single cell line, and a single set of assays, and then we're going to vary how we prepare that sample and watch what happens to the outcomes of the assay. So, as Pat mentioned, our objective is to investigate the effect of sample preparation techniques on the outcome of a standard assay panel. We're going to be using filter samples that were collected by the California Air Resources Board for a diesel engine. And we are also going to include a National Institute of Standards standard of diesel PM, as well as the filter blank. And as I mentioned, this is going to be from a single study of a chassis dynamometer. And we're going to keep a constant assay panel, and we're going to vary how we prepare that PM for that assay. Now, the null hypothesis is that it doesn't matter how you prepare the sample. You should get the same outcome regardless of the sample preparation technique. So that is the null hypothesis. What are our test hypotheses? Well, we came up with these before the study. These were a part of the proposal. So first, that the outcome of the assay is going to depend on the nature of the extraction. By the nature of the extraction, I mean, am I trying to do an exhaustive extraction where I want to pull the whole particle off of that filter? Or am I trying to do a selective extraction where I am only pulling certain compounds or certain components out of this complex PM mixture, and I want to look at the toxicity of those individual components free from each other? Number two, it's going to depend on what extraction solvents I choose to use. Do I use a polar solvent like water? Do I use polar A product like dichloromethyl? Do I use a completely non-polar like toluene, benzene, hexane? 3, hypothesize that we're going to see toxicological matrix effects. So what does that mean? I borrow this term from chemistry. And the basic idea is that the existence of one compound together with another somehow inhibits the detection of both. It can either be synergistic or it can be antagonistic. But the presence of both compounds together interferes with the detection of one over the other. So how does that relate to toxicology? The first is that if I go and I take a whole PM and I take different fractions and I test the toxicity of each of those fractions and I sum together that toxicological response, that somehow is going to be greater than the response from the composite of all those different fractions taken together. Another is that I said a toxicologically inert component together with a toxicologically active component can interfere with the detection of that component, either synergistically or antagonistically. And then lastly is actually a bioavailability, that perhaps the particle matrix is blocking the access of the particular active component from the biological system. And lastly is that the most robust, the most reliable sample preparation technique is going to be a function of the assay. So that means different assays are going to respond better to different sample preparation techniques than others. We'll talk more about that later. So the end deliverable of this project is supposed to be a standard operating procedure for PM sample preparation, which may be a function of the assay that you're looking at, and it may be specific to the type of particle that you're dealing with. In this case, we're dealing with diesel exhaust particles. So just some fundamentals to start off with. Sample preparation is everywhere throughout the chemical industry. It is composed of 3 parts. There's a pre-treatment, where the physical chemical characteristics are altered prior to the extraction. There is a solvent extraction, where components are separated from liquid, solid, or semi-solid matrix and dissolved in a compatible solvent. Then there's post-extraction cleanup, where the matrix residues or any interfering co-extractables are removed. And collectively, it is referred to as solid phase extraction, SPE, and it includes filtering, adsorbents, and ion exchange interfaces. So once decide on your pre-treatment, solvent extraction, post-extraction cleanup is all decided by what kind of extraction you want to do. And I've already talked about this, but exhaustive means that you want to maximize the number and amount of compounds that you're extracting from your filter. So for PM extraction, that means you want to bring the entire particle off the filter into solution, and you want to dose with that particle. The other is selective, and you want to maximize specific components and minimize the co-extraction of other unwanted material. So for PM extraction, this would be a fractionated PM, where you're taking different fractions of the whole PM and you're testing the toxicity of those individually. So quickly, we can go through some existing extraction techniques. Solvent extraction is a diffusion-based process. The solvent diffuses into the particle matrix. It dissolves components. It diffuses back out into the bulk solution. So it is affected by 3 main variables: solvent that you use, source of energy that you use, and the time you subject your sample to that. So heat reflux extraction uses heat as the energy source. What heat does is it increases the viscosity of the solvent, increases its ability to get into the particle matrix, dissolve the compounds, and bring them back out into the bulk solution. For those that don't know, reflux simply means that you put a solvent into a flask, you bring it to its boiling point, you let it boil off, and you have a condenser on top. And the condenser has a sheath flow around it of cold water, and it condenses those gases as they try to escape, puts them back down into solution, and you continually just reflux. You bring in it, boiling it, condensing, boiling, condensing. And you keep doing that for hours and hours and hours until you get your components out of your particle. It's very simple, it's very cost effective, and it's small scale. Next is a Soxhlet extraction. It's the exact same thing as a heat reflux, except it has inline filtration. So your sample is actually put into a little thimble, and as the refluxing vapors condense, it goes back over the thimble, which is actually a filter material. It goes across your sample and back into the bulk flask. Also, we've got sonication extraction. And sonication uses the ultrasonic energy from imploding bubbles in solution as the energy source to remove the particles from the filter. There's an increased extraction specificity, a decrease in extraction time with sonication. Another is microwave-assisted extraction, and actually use microwave radiation to heat up the sample. Again, the same principle applies here: increased heat, increased viscosity, increased extraction efficiency. It's very high throughput, it's very efficient, both in terms of time and solvent. Supercritical fluid extraction, predominantly CO2 is used. And at supercritical conditions, it has hybrid gas and liquid-like properties, increases diffusivity as well as viscosity, increases penetration. But there's special handling requirements. It is expensive, and there's limited applicability. And then last but not least is pressurized fluid extraction, is also referred to as accelerated solvent extraction. You put your sample into a cell, and you increase pressure, you increase temperature, and this decreases viscosity, increases your extraction kinetics. And this is one of the most commonly used methods in high throughput labs for chemical analysis. And it's starting as well as for high throughput toxicity screening as well. So on the right is a table just kind of showing, putting things in perspective about the amount of time each technique takes, as well as the amount of solvent that is used for each technique. And so it goes from a couple of days down to a matter of, tens of minutes, and from hundreds of mils of solvent down to tens of mils of solvent. So those are the existing extraction techniques. And so once you determine your extraction technique, you need to figure out what solvent you're going to use. So here's some common solvents. And the things we want to pay attention to are the boiling point and the polar bonds, as well as the hydrogen bonding. And there are three general classes of solvents: non-polar solvents, hexane, benzene, toluene; polar aprotic solvents. They're polar without the hydrogen bonding. For example, THF, dichloromethane, acetone. And then you have your polar protic solvents, methanol, most alcohols, water. So when selecting, can't see that one either, sorry. When selecting, you want to be wary of your boiling point because you have semi-volatile species in your particles. So at some point, you're going to have to remove the solvent. And when you remove the solvent, you need to be wary of what temperature you have to do that, else you're going to lose semi-volatiles. And then polar bonds, how polar is it? As well as hydrogen bonding, how much hydrogen bonding does it have? So you take all of these things into consideration and you do your selection. So what we chose for our studies was toluene as the nonpolar, dichloromethane as our polar aprotic, and then methanol as our polar protic. We chose these because of their very low boiling points, as well as this is benzene ring in it, and it has a great affinity for PAHs as far as being able to dissolve PAHs. So those are the solvents. Sample collection and extraction objectives. So obviously we're doing filter extractions, and we're using the chassis dynamometer setup of the Air Resources Board. There are numerous different types of collection substrates that we could use. In this study, we're using Teflon. And some extraction objectives depend on the study. So for in vitro studies, this is for animal models, we want to try to conserve the original physical and chemical properties of the PM so that our exposure represents true population exposure as much as possible. For chemical characterization, PM is chemically complex, so your extraction objective is try to remove the target compound while minimizing the co-extraction of other compounds. And then for in vivo studies, which we're doing for this work, assays are designed for specific endpoints, i.e. inflammation or reactive oxygen species. And the objective is try to pick the most robust and reliable response for a given assay. And in that light, the best extraction technique is likely to be assay specific. So our particulate matter source that we chose is diesel exhaust particles, the most heavily studied source of air pollution, dating back since the 1950s, I believe, yes? The US EPA began a research program in 1979. There were the chronic exposure studies in the 1980s, followed by the characterization studies in the 90s and the 2000s. Anything from stationary, mobile, roadside tunnel, dynamometer, laboratory generated. And the impetus was the detailed physical and chemical characterization, mainly emission factors and rates. Since then, there's been an exponential increase in sophistication, scope, and resolution of toxicity testing. As far as assays, reverse transcription, polymerase chain reaction, PCR, new animal models, both transgenic and knockouts, new cell lines, human lung, macrophages, stem cells, new endpoints, new injury metrics, as well as new exposure scenarios. So if you go through the literature and you try to look back and see some of the common diesel exhaust particle extraction techniques, this is what you see. So liquid impingers are used in some cases, in which case the fluid flow is drawn directly through a liquid and the particles are suspended directly into that liquid, and then you concentrate the solvent and you dose your assay. For dynamometer studies or dilution sampling, you get whole free particles that are scraped off of some part of the system, usually the heat exchange. Or you can get filter deposits, which we're using in this case. We're using both in this case, actually. And you can do either an exhaustive or selective. And common solvents that are used are dichloromethane, methanol, hexane, benzene. Common extraction techniques that are used are agitation vortexing, sonication, and pressurized fluid extraction. Some of these use a single solvent. Some of them use multiple solvents. Some are done in parallel, some are done in a series. There's a bunch of different post-extraction cleanup, mostly filtration, and then solvent removal, and then resuspension in whatever delivery vehicle you're going to use, followed by dosing your assay. So given all of those possible choices, take 3 samples, 6 sample preparation techniques, and a 5-point assay panel. So the samples that we're going to use are, like I said, the chassis dynamometers from the CARB heavy-duty engine emissions testing laboratory using a 2000 Freightliner truck with the 2000 Caterpillar C15 engine, running on California certified ultra-low diesel fuel. They used an urban dynamometer driving schedule. Samples drawn directly from a dilution tunnel onto a Teflon filter sheet using a high-volume filter sampler. Collection time was about 35 minutes, or about 11 miles, which relates to 2 back-to-back UDDS cycles. We also have a filter blank from the chassis dynamometer study, and then we also have a NIST standard, SRM 1650. And the abbreviations I'll be using throughout are dyno depth for the chassis, FB for the field blank, and NIST depth for the NIST standard. So those are the 3 samples. The 6 sample preparation techniques I chose are 2 exhaustive. These are the whole particle extractions. I'm going to use a single solvent sonication extraction using water, and then a serial multiple solvent sonication extraction, which is going to use 3 different solvents in series, including dichloromethane, methanol, and toluene. Those 3 separate sonication extracts will be reconstituted into a single extract, and that will be used for the assay. And I also chose 4 selective or PM fractionation extractions. I did a single solvent heat reflux using each of the chosen solvents individually. That's dichloromethane, methanol, and toluene. as well as a serial multiple solvent heat reflux extraction, where again, I'm going to use each of the solvents independently, and then at the end, put it all back together into one extract. And I did independent replications of each of these 6 techniques, and I pulled them together into a single extract, so that that single extract inherently includes any kind of systematic errors in the sample preparation. So that leads us to the assay panel. Use 937 macrophages plus qPCR. We looked at reactive oxygen species production. This is an acellular metric, and it studies the ability of the PM itself to produce ROS. Inflammation is measured by cyclooxygenase 2 and interleukin 8. Cellular response to PAHs is measured by the expression of cytochrome P451A1. And then mutagenicity was studied by the Ames assay. We did a 3-point dose response curve for each combination of assay and extract, and then we repeated in duplicate at a linear part of the dose response curve. So that's the experimental design: 3 samples, 6 sample preparation techniques, and a 5-point assay. And there's the measurement matrix right there. The assay is on the far left column. This is ROS production, inflammation, PAH response, mutagenicity. Across the top, we have 1, 2, 3, 4, 5, 6 different extraction techniques. Water sonication. DMT stands for dichloromethane, methanol, toluene sonication extraction. DCM is dichloromethane heat reflux extraction, methanol heat reflux, toluene heat reflux, and then again the 3 in series: dichloromethane, methanol, and toluene. Gives 3-point dose response as well as the test dose for each of our assays. Here's just a quick picture of the extraction apparatus in the lab. Here, oops, sorry. This is, does a bunch of stuff. This is in distillation configuration, where the distillation column is condensing vapors that are being boiled out of solution and down and collected in the still. You can flip this condensation distillation column up, right, and then it becomes the reflux apparatus. Also back here, I have vacuum lines for vacuum distillation. I also have nitrogen lines for nitrogen blowdown. So pretty much everything is happening in this little fume right here: the heat reflux, the removal of the solvents, and the final sample. On the right is a picture, wrong button, of the syringe filtration. So that's the post-extraction cleanup step, is I actually remove the co-extractables, remove the particle matrix from the selective PM fractionation samples. And I use a syringe filter. It's stainless steel, 200 nanometer pore size Teflon filters. And the weights on top just give constant pressure on a glass syringe. And it pushes it through the syringe filter holder here and into my collection flask. And extraction results, it's a hard picture to see, I apologize. Up at the top is the original filter sample, and below it are pictures of different extracted filters. This is from the dichloromethane reflux extraction. The top one is the filter piece after extraction. The bottom one is the actual post-extraction filtration filter that collects the co-extractable residues and matrix. Second one is the methanol. Third is toluene. So these are all the heat reflux extractions. On the right, these are from the sonication extractions. DMT, again, is the 3 solvents in series. That's dichloromethane, methanol, and toluene. And then water. General observations that extraction efficiency, which is the amount of mass that is used, removed from the filter, generally increases going from, or decreases going from dichloromethane, has a higher extraction efficiency than toluene, greater than methanol, greater than water. And you can see over here that this filter piece that was extracted with dichloromethane looks almost clean. It's almost white. Whereas the other ones still have stained remnants of particulate matter on there. And then you can also see a significant amount of matrix is still left behind after the post-extraction filtration step. And in general, the heat reflux is much more efficient than sonication for diesel exhaust particles on filters. And lastly, that the post-extraction cleanup is moving about 30 to 60% of the extracted diesel exhaust particles are removed by the post-extraction filtration. So those are general results from extraction. So now we can jump into the toxicological data. The first one I show here is SIP1A1. This is response to PAHs. Y-axis shows the expression level. This group right here is all of the dynamometer depth. And this cluster over here is all of the NIST standard. And DCM, dichloromethane heat reflux, filled blank next to the corresponding sample. And the same for methanol. And the same for DMT, which again is dichloromethane methanol toluene. And the same for the water sonication. The field blank is next to the sample. And lastly, for DMT sonication extraction. And the A means that the sample is statistically significant compared to the field blank with a P of 0.05. There are no field blanks for the NIST, so these are all significant. The general observations are that SIP1A1 is significantly upregulated by all of the extracts relative to vehicle control. NIST standard expression is greater than the dynamometer depth for a given extraction technique across the board for all extraction techniques. For both the dynamometer and the NIST standard, the heat reflux extraction is always greater than the sonication extraction. The combination of 3 in series is greatest, and the combination of 3 solvents in series for the sonication is much larger than the water sonication. So those are the general observations, and we're going to see these repeat again as we go through the different assays. So next is, oh yeah, so I wanted to show you this. Oh, you can see it here, good. Unbeknownst to Norm or Chris, I actually repeated several of the extractions, and I threw them in as duplicates. This is the metric of repeatability. So that's what you're seeing here. There are 2 dichloromethane heat reflux extractions side by side, as well as 2 methanols. This was a blind study. They did not go with that going in, but this just gives a metric of repeatability to show that what we're doing is repeatable. And you'll see this again as I go through the data. So here is interleukin 8, and this is a metric of inflammation. Same setup. Expression level on the y-axis. This cluster is for the dynamometer. This cluster is for the NIST standard. On the dynamometer, you have the field blank next to the actual sample all across the board. A means that the sample expression is significantly different than the field blank. So again, we're seeing a lot of the same trends that we saw for SIP1A1, most notably that the NIST is consistently greater than the dynamometer, and that for both the dynamometer and the NIST standard, the heat reflux is significantly more potent than the sonication, and that the 3 solvents in series tends to give the largest response, and that for the sonication extraction, the 3 solvents in series is greater than water alone. One additional observation here is that for the dyno, the water sonication was not even significantly different than the field blank extract, and I will explain the significance of all of these continuing observations towards the end. I'm going to continue with TOX2. Again, all extracts are significantly above a vehicle control, but in this case, only the dichloromethane heat reflux and the 3 solvents in series were significantly different than field blank. Everything else was not. Again, we're seeing the same general observations. The NIST for a given extraction technique is always greater than a dynamometer for the same extraction technique. For both the dynamometer and the NIST standard, the heat reflux is eliciting a greater response than the sonication. Solvents in series for the heat reflux tends to give the largest response. For the sonication extraction, the 3 solvents in series is always greater than the water alone. Moving on to dose response. So like I said, we did a dose response for each of the assays using 3 different doses, and this shows the results for CYP1A1 for a selection of different extraction techniques, expression on the y-axis, the different extraction techniques on the x for a dynamometer sample. The rest of these are the NIST samples. The 3 different doses are 1 1/4, 2 1/2, and 5 micrograms. And we see a very nice dose response for all the different samples. And again, you see good agreement for this repeat sample that I did. The methanol heat reflux is repeated here. So this gives you a good sense of repeatability in our experiments. And it's actually quite nice. Here's the same exact thing for interleukin 8. Expression on the y, different extraction techniques on the x. 1 dynamometer, and the rest are the same doses, 1 1/4, 2 1/2, and 5. And again, we see good dose response. Here, the largest goes smaller, but it's not statistically different than the middle dose. But everything else, same here. The largest is not significantly different than the middle. But still, you see the general trend of increasing expression with increasing dose. And finally, this is for COX-2 dose response. Expression versus extraction technique. Same 1 1/4, 2 1/2, 5 microgram doses. And again, we get pretty good linear response for all of our different extraction techniques. And here's the repeatability. These match up very nicely. They're both the methanol heat reflux, done in duplicate, done blinded, and they look very nice next to each other. So now, move on to mutagenicity. On the left plot is all of the dynamometer samples. On the right plot are all the NIST standards. Y-axis gives us a metric of response. The x-axis gives us the different extraction techniques. Again, we've seen the same general observations we saw for the other markers. The NIST for a given extraction technique is significantly greater than the dynamometer study by an order of magnitude. Flux extract activity is always greater than the sonication extract. A DMT, which is the 3 solvents in series for the sonication, is always greater than water alone. And the water sonication was not significantly different than the field blank control for the dynamometer study. Next, and finally, we have the DDT assay, which is reactive oxygen species production. Same setup as the previous slide. On the left plot is the dynamometer samples, and on the right are the NIST standard. Y-axis gives us response. The x-axis gives us the sample preparation technique. And both the sample as well as its corresponding field blank are shown for the dyno. And in general, we see that for the dynamometer study, the DDT consumption rates are actually quite low. And in fact, the methanol heat reflux extraction of the field blank turns out to be the largest response. And the data is just generally noisy and not statistically significant, so we can't say a lot about reactive oxygen species from the dynamometer studies, and we think that maybe we just did too low of a concentration on those assays. But for the NIST standard, much cleaner data, and the methanol heat reflux extraction gives the largest response. But we have a bit of a repeatability issue here for the dichloromethane. This is the one I did in repeat, and they're significantly different from one another, so I'm not sure what's going on there. So we're going to go back to the test hypotheses that I laid out at the very beginning and try to evaluate these based upon the data that I showed. First and foremost, outcomes are going to be dependent upon extraction technique, whether it is exhaustive versus selective. And this is one of the general trends that I kept alluding to as I went through. And in every single assay, we saw that the heat reflux extraction was significantly greater than the sonication extraction. So the heat reflux extraction is selective technique. It's going in and it's removing certain classes of compounds that tend to be soluble in the solvent of choice, whereas the sonication is exhaustive. It includes everything on the filter. There are no post-extraction cleanup steps. So what this is saying is that somehow selective extraction of individual components is amplifying the effect by removing inactive components, whereas somehow the particle matrix is interfering with the toxicity of the active components. And if we go in there and we selectively remove those active components, we're seeing a lot larger response than if we tried to measure everything together in the same particle. So that's a major finding of this, and we hypothesized that that would be the case in the beginning. Secondly, outcomes are dependent upon the solvent system. And again, I tried to show this consistently through each of the different assays, and we observed it in both the dynamometer samples as well as the NIST standards. And we saw that for sonication, the series of different solvents was consistently greater than water alone, suggesting that perhaps it is the polar or nonpolar components that are eliciting the effect. CYP1A1 was greatest for toluene, which may be most effective at solvating PAHs, which the CYP1A1 is reacting to. DDT consumption was greatest for methanol, suggesting that perhaps it is the polar compounds, such as the oxygenated organic compounds, that are driving reactive oxygen species production at the particle level. Thirdly is the existence of toxicological matrix effects. And I said that the component sum was going to be greater than the composite. And in fact, we did see this. You can't see it on an equal mass basis, but if you go back to the dose response curves for the heat reflux extraction, you'll notice that if you add the response from the three independent extracts, dichloromethane, methanol, and toluene, at the very lowest dose, and you compare that to the combination of all those three solvents put back together into a single composite, you will notice it is significantly larger, suggesting that there is some kind of composite interference where the combination of components together in a mixture is interfering with the response to individual components alone. And lastly, robustness, repeatability, reliability of each technique is going to be dependent on the assay. And again, we saw this consistently throughout. Two examples I gave previously. DDT consumption is greatest for the methanol extract, where CYP1A1 was greatest for the toluene. And there's a lot of different examples of this throughout the data, but solvent system and the extraction method that you use is going to affect the outcome of the assay, and it depends on what assay used, and it's probably also going to depend on what type of particulate matter you use. So, in conclusion, for a given extraction technique, response to the NIST standard is significantly greater than to the dynamometer standard or study for all of the assays, for all of the extraction techniques. And we have attributed this to compositional differences and the diesel particles that we used. For NIST, they were collected way back in 1984 by several different uncontrolled diesel engines. They scraped the samples off the heat exchangers, a high sulfur and aromatic content of the fuel, whereas for the dynamometer study, we used a single modern year 2000 diesel engine. We sampled onto filter from the dilution tunnel, and it was an ultra-low sulfur content fuel. Secondly, heat reflux extraction consistently enhances the response of active components by removing inactive matrix components that are included in the sonication or whole particle extraction methods. Non-polar compounds seem to elicit the greatest response across all assays, with the exception of reactive oxygen species, which seems to be dominated or driven by the more polar components of the diesel exhaust particles. There is strong evidence for the existence of composite interference or toxicological matrix effects. And lastly, although different techniques appear equally repeatable across assays, those eliciting most robust response are assay specific. So, in closing, making recommendations. First and foremost, the chemical characterization was not included in the study, but would be insightful and informative to know what chemical differences are driving the observed differential responses in the toxicological assays. And continue to explore the relationship between the complexity of PM composition and how this affects toxicity outcome. Selective versus exhaustive extraction. This is a continuing conversation that we're having. What is the more appropriate metric to measure toxicity outcomes? You could argue that the whole particle is what the human is exposed to on a daily basis, and that should be what you were using to determine toxicity. The other side of the argument is that we don't know the bioavailability of the different compounds in a very complex particle mixture, so we need to go in and we need to separate them out and we need to test the toxicity of them one by one individually, and we need to take that approach. And this is an ongoing discussion about what is the more appropriate evaluation metric if we want to look at the toxicity of a very, very complex system, not only at the particle level, but at the exposure level, how people are exposed, when they're exposed, vicinity, daily routines, activity patterns, becomes very complicated very, very quickly. For standardization, this was supposed to be an end deliverable of this project, so my recommendation is that we want a single method for all assays. Dichloromethane heat reflux extraction seems to be the most efficient and the most potent across the board. If you want assay-specific methods, then my recommendations would be a methanol heat reflux for the DDT ROS. It would be the dichloromethane heat reflux for the molecular markers and mutagenicity. And it would be a toluene heat reflux for CYP1A1. If we decided that the exhaustive approach, the whole particle approach, is the appropriate metric to use rather than the selective approach, then my recommendation would be first to go back and compare the heat reflux method without any of the post-extraction filtration cleanup methods to the sonication extraction and to see how they compare before making a decision. And a couple other tidbits of information is that if this is to be repeated, we were very limited by the amount of filter sample that we had available to us, and it affected the outcome of our study, and I would recommend that there be ample material available to run everything in high mass loadings rather than very low. We were at the hundreds of microgram level where we should have been closer to the milligram level in dealing with this as far as quantitative transfer, gravimetric analysis, all these things make a difference. So if we're going to do this again or going in the future, I would recommend going with a higher mass loading on the filters. Very careful about your labware selection. This was a very unexpected part of this. When we're going through and trying to characterize all of these new techniques and we're using different kinds of labware, we saw a lot of contamination. depending on what kind of materials you chose to use in the lab. And I spent months and months and months trying to find the best materials, and it all came down to stainless steel and glass, Teflon. So that was a hard lesson learned, is that the labware selection is a very critical part of the entire process to minimize any kind of interference. And with that, I would like to open the floor for questions and to put up our email addresses if you would like to email us questions instead.
I guess we'd like to open up the floor for any questions in the room.
Yeah. Okay. Yeah, thank you. Very nice presentation. Just a clarification about the masses used for the NIST versus the depth. They were approximately the same as you were going through all your different procedures?
They were the same. The difference I can measure it out in a high quantity, to a high degree of accuracy, because your balance that you're using
Mhm.
so your more precise is higher, your less precise is lower. I have more faith in the But with that being said, everything was measured at a dry mass. All of the concentrations of stock solution.
Okay. So initially they were about the same, but you also showed with the different extraction procedures, they had different efficiencies in terms of extraction. So when you went on to the next step of testing, using your different assays with the different extraction yields, did you somehow try to make those equivalent, or?
So the mass that I use. Oh, okay. So the mass that I used were for the final extract. So after all of the intermediate post-extraction cleanup steps are taken, I weigh the dry mass of the extract, and that is the mass that is given to Chris and Norm to make their stock solutions. So everything, it's not the starting mass, it's not the mass on the filter, it's actually the extracted mass that I measured directly
Got it.
to make
Okay.
sure that it is normalized out and we're not doing a dose response inadvertently
Great.
when we're trying it.
Thank you.
Hi, Keith. It was a good presentation. Thanks. I'm wondering about the NIST standard and the high sulfur content of the fuel. I know that wasn't the focus of this. You're interested in the biology, not the chemistry. But what do you think that high sulfur content of the fuel might be doing that would cause the response to be higher? If I invite you to speculate, since, like you said, you don't have the composition data.
I was thinking more along the lines of the high aromatic content that might have more of an influence than the sulfur content. I know that during the combustion process, some fraction of sulfur in the fuel is directly converted to sulfur-3, in which it becomes oxidative as forming sulfuric acid or other kinds of damaging. But other than that, I have no idea why sulfur content would affect biological outcome. I was thinking more of the aromatic content of the fuel would have more impact on the pH concentrations. Also, there's the issue of combustion efficiency between the different engines. The newer engines have a higher efficiency, which means you're going from the longer chain alkanes to the shorter chain on the output side, and most of those are going to be in the gas phase and not the particle phase. where the inefficiency of the older engines is going to produce more PAHs that are going to remain in the particle phase. But I don't have a good answer for the sulfur content.
I also want to thank you for a great presentation. I want to ask you a higher level question. You know, I came here looking for some final answers, and I don't think we're any close to that, because I hear your call for more mass so you can do better extractions and sampling. And unfortunately, the problem we have is pulling us in the opposite direction, meaning a 2000 model year engine is not what I would call a modern engine. A modern engine is one that has advanced controls, which is probably going to result in 99.9% reduction in PM. So the trend is, as you look forward and the Air Resources Board looks to apply the findings, the challenge is going to be that you're not moving to more mass, you're moving to less mass. So my question for you is this. In the world of toxicological analyses, extractions, are there any trends for new techniques, for new ways of doing what you've done here, you know, that are not going to be so reliant on more mass collection? What are the trends in your industry, for that matter?
So I was just up at Oregon State University giving a talk. And they have, you know, state-of-the-art zebrafish facility out there. And it's one of the new animal models that people are using for toxicological analysis. And they run all kinds of different samples, and I've given them specific samples to run. And one of the methods they use is accelerated solvent extraction. And so this is the direction of high throughput extraction for high throughput toxicological screening. And so basically, there's a cell, you put your sample in, you put your solvent in, you put it into an instrument, raises the pressure to 2000 psi, raises the temperature to 200 degrees Celsius. 15 minutes later, boom, you're done. You have inline filtration and outspits, and you put it into your assay, you put it into your animal model. And I was having a one-on-one with one of the girls up there that runs the facility, and she was showing me some data from she was using dichloromethane, and she was actually getting higher toxicity out of the blank than she was out of the filter sample. And it was so bad that it was killing the zebrafish. And now this is supposed to just to be a state-of-the-art, off-the-shelf, accelerated solvent extraction with a very common solvent, dichloromethane, and getting a very, very weird response in the animal model that they're using. And so I would caution against moving in one direction too quickly without covering your tracks. A lot of the stuff that I use has been tested for decades. Toxicological extraction has been around. Heat reflux has been around. And I chose those methods carefully, but they are more labor-intensive, time-intensive, solvent-intensive. There's a lot more chance for losses along the way from beginning to end. And I understand that engine technology is going towards lower, but my contention would be that you could just sample longer to get more mass. And I would also contend that it's not clear that filter collection, extraction, acute bolus exposure is the appropriate way to go. There's also real-time ambient exposures, doing exposures out in the field where you're actually looking at a real vehicle fleet mixture. Modern day, it has the old, the new, the electric, the hybrid. These are changing, these are evolving. The exposure to traffic-related air pollution is going to change and evolve with that changing vehicle fleet mixture, and we need to be able to track how that is affecting human health. You know, there's two sides to the coin. There's reducing emissions for environmental impacts, such as climate change, but there's also reducing emissions and changing the composition of those emissions for human health. And it's not clear we're going to be able to answer that in the laboratory alone. We are going to need real-world systems that we can use to dose our animals with or to dose our cellular systems with to get a real answer to how it's going to change. A single dynamometer study is informative, but I don't think it's the whole picture.
Okay, do we have any other questions in the room? I just have a follow-up question, I guess, to Alberto's question. As engines get cleaner, obviously we're going to get less PM from them, but how adaptable would this be to more ambient sources, newer type sources as they come along? Do you think we could still use information from this project for like, ambient air study that we're running right now?
Yeah, absolutely. Anything that you can collect on a filter, you can do an extraction and a high throughput assay and get some good answers about relative toxicity, as you mentioned in your opening slides. We don't know exactly how to correlate absolute toxicity back to human health yet, but relative metrics are very useful in their own right as far as setting mitigation strategies and knowing that this source is more or less toxic than that one, then you can be more careful when you're around this source, or perhaps you can regulate this source more than a different source. And that's been an argument that's been around for a long time, is that not all sources of PM are created equal, right?
Right.
Is sea salt spray as toxic as diesel exhaust? Probably not. Are they regulated the same? Yes, they are. That costs industry and consumer quite a bit of money. If we could be smarter about the way we regulate our sources, then maybe we could save more lives. We could decrease the burden on regulatory agencies. We could decrease the burden on consumers and industry. So,
Okay, thanks. We did have one question just come up from Mary Beth Swire at ARB. And I think maybe someone else will be able to answer this. Any test data or plans to do so on biodiesel exhaust? Any of you two?
Well, with UC Riverside and also with the ARB and some other, in conjunction with a lot of people, there was a major study to do a lot of biodiesel in different types of feedstocks, basically soy, animal, and also what they call a renewable, a new type of hydrotreated biodiesel. So that's in the writing stage right now. It's been tested.
Thanks, Norm. Okay, we just have one other question just come up from, let's see. They don't say which one, but her name is Laura Rosenberg Hyder. It's just a series of questions. And maybe easier if you can answer these later on. Which method is best for VOCs, NOx, metallic dust, trimethylene? Okay, she basically has a list of counts. Any quick answer to that, or maybe we can take this off?
sure. First,
Level.
we got to distinguish between gas phase and particle phase. So the first few items you had on your list would be VOCs and NOx.
Yeah.
And those are gas phase and
I guess metallic, actually, I was kind of curious about metals as well. Which,
metals, typically the way you remove metals is by acid digestion, and you're automatically changing the composition. And the other thing about metals is they typically form the seed of the particle, which means they're at the center of the particle, and they're typically encapsulated by layers of organics or inorganic condensed salts. So typically what you want to do is you want to remove those layers to expose the metal core, and then you could do an assay on the metal core. For example, you could do a simple water sonication followed by a liquid-liquid extraction and some kind of organic solvent to remove any organics, and then whatever you have left in the water phase would be a metal core particle that you could then run a series of assays on.
okay, if there are no more questions, I'd like to thank the speaker and his co-investigators again. Asking like formaldehyde, and I just realized a lot of things. Those ones survive your extraction.